Today I will explain it. In addition to this, I will also explain the types of muscle contraction. There are two types, isotonic, isometric. What are the difference between the two? So that I will be discussing. Then we will also discuss the skeletal muscle needs energy, ATP. We have seen. Now what are the energy sources for muscle contraction? How the muscles get their energy, means how they produce their ATPs. Because all the functions we will be able to see, the ATP is very, very essential thing. That's why the muscle cells are having lot of mitochondria in it. So that's why generators are there to produce ATP. Now what will be the raw material? What they will utilize? Whether it is carbohydrate, or it is fat, or it is protein, or something else. And what will be the mechanism? Whether it is anaerobic or aerobic or any other mechanisms. So we'll discuss that one. So we'll go through this first. So in addition to that, to this, as I told you, that will be discussed. So now I am coming to this. There is no need of explanation of this. Also, you have gone through it, so no need of any explanation. See, cells are surrounded and bundled by connective tissue. We have seen there is perimysium, endomysium. So they provide, when muscle will be contracted, lot of tension will be generated. Now this tension are to be, otherwise the cells fibers may be damaged. So it is protected by this connective tissue. Epimysium, perimysium, endomysium. All these are connected to tendon. So there are several muscle proteins. There are actin and myosin. And there are some regulatory proteins, tropomyosin, troponin. Troponin has got three components. One is, this is TNC or TROP-C, T-R-O-P, TROP-C. Instead of TN, we will write TROP, TROP-C, TROP-P, TROP-I or TROP-A. So what are their functions? TROP-A or TROP-I, this troponin component, they will bind the actin. They will remain fixed with the actin. And TROP-C, it will bind calcium ion. And another TROP-T, it will bind tropomyosin thread. There is tropomyosin thread, so it will bind with this. So these three components are necessary. This is thick filament, means myosin. So these are the different components of thick filament. What is this? There are heavy myormyosin and these are the light myormyosin. Heavy myormyosin has got two chains. These are both the chains are spirally arranged. At one end, they are not straight or spiral. They are separated and they can be bent, flexed, hinged. There you can think about a hinge there. So with this it can move like this. With this, because of this mechanism, they will make the power stroke. Okay. These are the myosin heads, and these are light chains. There are two light chains. One is alkaline light chain, another is a regulatory light chain. Okay, it is clear to you? Fine. And about the actin filaments, we can see there are three components in the actin. There are globular proteins, beaded, two beaded structures, two beaded chains are there. They are also spirally arranged. And another thread-like structure, this is tropomyosin. They also spirally arranged over the actin filaments. In addition to these, there is troponin. And as I said, troponin has got three components. These are very essential for muscle contraction, for EC coupling. So this is the anatomical structure of the actin and myosin, contractile proteins. Go through this. If any point need to be explained, I will explain. This is the component of actin. So every actin globule has got an active site, myosin binding site. The myosin head will bind with this site, will attach to this site, then only it will produce the swivel movement or power stroke. This is how they are aligned, and this is the distance 70 nanometer. Here you will find the M line. In M line, the myosins are from both sides. The myosin myosin, they are attached to this here. During muscle contraction, they will not shorten. Actin will also not shorten. Only there will be sliding. These filaments are not shortened. They only slides. That is explained by, what is the theory? Sliding filament theory. In the myosin head, there are light myosin. So these are the components of light myosin. What are these? Regulatory light chain and alkaline light chain. What is its function? Stabilizing myosin head. Its function is to stabilize the myosin head. And what is its function? Regulatory light chain. Regulate ATPase activity. Regulate ATPase activity of the myosin head. Is it clear? Fine. This is the arrangement. Myosin, Z-line, actins. So during muscle contraction, they will be coming closer. As a result, there are different segments. This segment will not be shortened. They will fix. Only this area will be shortened. Since they will be coming closer, because of this cross-bridge formation and contraction, both these actin filaments will be coming closer. As a result, these band will be shortened. These are what? Actin and myosin, muscle fibers. These are within the muscle fiber, there are actin and myosin. So these are actin and myosin. So now what happens? This is the cell membrane of the sarcoplasm, of the myocyte. Myocyte means muscle cells. It may be myocytes, it may be small to smooth muscle, it may be cardiac muscle, can be skeletal muscle cells. So by myocyte means we want to explain muscle cell. See, from these sarcolemma, this sarcolemma folds. This sarcolemma is folded. Now this folding is inserted within the sarcoplasm. Suppose this is the sarcolemma. So this sarcolemma will be folded. These will be folded and they will be coming within sarcoplasm. And they will surround the myofibrils. So these are T-tubules. See there, structure is like alphabet T. Sarcolemma and this tube inserted within the sarcoplasm and forming a T shape. That's why we call it T-tubule. They, after inserting within the sarcoplasm, they come close to endoplasmic reticulum. But here, in case of muscle cell, we will not call them endoplasmic reticulum. What we will call them? Sarcoplasmic reticulum. So there, they will be coming closer. T-tubules and sarcoplasmic reticulum will be coming closer. Sarcolemma, T-tubule. So this is actually sarcolemma. Now they will be coming close to endoplasmic reticulum. This is simplified. Endoplasmic, sarcoplasmic reticulum is simplified structure. This area forms a triad. Three components are there. Three tubular structures are there. 1, 2. These are part of sarcoplasmic reticulum and this is part of endoplasmic reticulum. So they actually create magic. We'll see what are the magic they create for muscle contraction. Here you will find, in the triad you will find numbers of receptors. And you have to remember these receptors. I will explain one after another. In this endoplasmic reticulum, lot of calcium ion. You will find lot of calcium ion. Because, so they are not, these endoplasmic reticulum, the content of the endoplasmic reticulum are not directly exposed to sarcoplasm. Because we know in the sarcoplasm, calcium concentration is very low. If its concentration is increased within the sarcoplasm, there will be muscle contraction. So we don't want every time muscle contraction. Otherwise, I will not be in a resting state. So normally there is, you can say, very minimum or no calcium ion within the sarcoplasm. If it is released from the sarcoplasmic reticulum, again after their action, they will be again coming back to the endoplasmic reticulum. We'll see how it is done. This is ECF. Outside the sarcolemma, there is fluid. There is ECF. ECF may contain calcium ion. ECF may contain calcium ion. So here also you will get calcium ion. Here also you will get, in the T-tubule, you will also get calcium ion, but not in sarcoplasm. Remember this. What is this? This is a nerve fiber, or this is the sarcolemma. This is the nerve endings, synaptic knob, motor end plate, and this is sarcolemma. What happens when action potentials come here through formation of loops? They will be coming here in the presynaptic membrane. When they will arrive at the presynaptic membrane, when this action potential through loop formation, current loop formation, they will come here. This is what? Active zone. We have explained earlier. This is the active zone. When this action potential will be coming to the active zone, so their calcium channel will be opened. Now, calcium will enter from ECF into this, and they will cause a release of they will cause movement of vesicles to the active zone. So, from here, the vesicles will move to this, and here they will fuse, and the content will be exocytosed into the synaptic cleft. Now, this acetylcholine will bind to a receptor. Acetylcholine receptors. Let me draw a bigger picture. Motor end plate, synaptic vesicle containing acetylcholine. So, action potential when will be arriving here, calcium ion will enter. This calcium ion will combine with calmodulin, and they will cause, this complex will cause release of the vesicles from the skeleton, cytoskeleton. Now, with the action of snare proteins, they will be shifted here, and the content will be released. Okay? So here what we get? Receptors. What are these receptors? Acetylcholine receptors. Now this acetylcholine, what are these? These are ACAs, acetylcholine. Now they will bind with receptors. We explained there are 5 proteins, 2 alpha, 1 beta, 1 gamma, 1 delta. The acetylcholines will bind with the alpha monomers. As a result, there will be conformational change. Within the channel, within the receptor, there is a channel. This channel is for sodium ion. Now the sodium ion from ECF, in the ECF, what is there? There is sodium ion. After acetylcholine binds with the receptor, this channel is open. Now sodium ion will enter within this. As a result, inside is negatively charged, here inside is negative charge, outside is positively charged. If sodium ion enters within the sarcoplasm, the inside will be gradually positive. It will lose negativity. So after some time, it will attain threshold value. When this threshold potential is attained, what will happen? All the voltage-gated sodium channels will open. Before that, it was not voltage-gated. It was the calcium, that is the acetylcholine channel. Within the channel, there is a sodium channel. So sodium will enter slowly. When they will cause, this sodium will cause changes in depolarization, and when the depolarization is reached to the threshold level, then what will happen? There are lot of voltage-gated channels. These channels will be opened up, and now lot of sodium ion will be entering, and action potential will be generated. Now we know these action potentials are propagatory in nature. They can propagate. So, like this, we have seen they form a loop. So this sort of loop, current loop, they travel. So here also they will form this sort of loop, current loop, current sink. So with this formation of sink, this action potential will be traveling along the sarcolemma. While traveling along the sarcolemma, so same action potential will be traveling. While traveling along the sarcolemma, it will get T-tubule. So it will enter within the T-tubule. Are there any confusion? Any question up to this? Right. Okay. Now this action potential is entering. When they have entered here, what will be there? This is T-tubule. Here, there will be number of receptors. DH, what is this? Dihydroxyridine, pyridine receptor. They are mechanically connected. You think of a spring. They are mechanically connected to another receptor. This is called RY receptor. Write it. Rhinidine. Rhinidine receptor. This is a receptor for calcium release. Within this, we said there are a lot of calcium ion. So this is the route. When this will be opened, this calcium ion can be released within the sarcoplasm. So this part is sarcoplasm. What is this receptor? Dihydroxy pyridine receptors. So when this action potential comes here, binds to this receptor, the receptor, when in presence of action potential, this receptor make a conformational change, makes a structural change. Because of this structural change, this receptor will be displaced from here, or it will open a channel. And this is calcium channel. Now what will happen? Because of arrival of action potential here, these channels are opened and they will cause release of these channels. Now what will happen? Lot of calcium ion will be entering. Now, they will also cause here the calcium ion is there. Here also there calcium ion. So from here calcium ion will be released. From here also some calcium ion will be released. But more amount will be coming from here. Here the amount is less. Now what happens? What happens when action potential arrive at the sarcotubular system? We'll tell. When action potential arrives here, what will happen? There will be changes in DH-PR channel. And they will cause opening of ryanidine channel. They will be displaced. Now, this calcium release channels is open. Here concentration of calcium ion is high, here it is less. So, they will come in here. Here this channel is also open. Some calcium ion will also be released. So this way we can see arrival of action potential in the T-tubular system causes a release of calcium ion from the T-tubule as well as from the endoplasmic reticulum into where? In the sarcoplasm. See, one question may come. Describe EC coupling. What do you mean by EC coupling? This is, see, excitation. Excitation by the action potential. So how action potential causes release of calcium ion from the cistern. These are nothing but cistern. The swollen part is cistern. The release of calcium ion from the cistern. Now what will be there? This calcium ion, release calcium ion will initiate muscle contraction. This process, excitation, release of calcium ion, initiation of muscle contraction. This process is called EC coupling. Now we have seen calcium ion is released. Now let's see what calcium ion will be doing. What is its role? Let's see. How many receptors you got in the triad till now? 2. One is DSPR, another is RYR, ryanidine phosphate, ryanidine receptors. Okay. There are 3 more receptors, channels, receptor channels. And their function is to, with the rest of the 3 channels, what will be their function? Reuptake of calcium again into the sarcotubular system and into the endoplasmic reticulum. We'll explain later. Here we'll find, here, some other receptors, channels. These are called sarca. S-E-R-C-A, right? What is sarca? S for sarcotubular. S stands for sarcotubular. ER stands for endoplasmic reticulum. C for calcium. A for ATPase. This is the full form of sarca. S-E-R-C-A. Can you repeat? Go, tell. Sarcotubular, endoplasmic, ER, calcium, ATPase. What is their function? See, ATPase means there will be breakdown of ATP. Means reuptake of calcium. You will be requiring ATP. So these are present here. But in the this is what? T-tubule. So some calcium ion was also released from here. So they are to come back. How they will be coming back? Again, there are 2 receptors. One will be an exchanger. Sodium calcium exchanger. Calcium will, from here, it will be entering here, and sodium in exchange, sodium will be coming out. So there will be an exchanger. What is the name of this exchanger? Sodium calcium exchanger. Their function, reuptake of calcium ion from sarcoplasm into the sarcotubular system, means into the ECF. Within this there is ECF. So this is one exchanger. Other than this, there is another receptor. This receptor is, there is another channel. This is what? Exchanger. This is membrane. This is the membrane. So membrane bound calcium ATPase. Membrane bound calcium ATPase, or also called plasma membrane. More precisely, it is plasma membrane. Plasma membrane calcium ATPase. So this is this protein. Here also ATP will be needed. Actually, what happens? The calcium is pumped in by this enzyme. What is this enzyme? What is this receptor? Plasma membrane calcium ATPase. Okay, so these, how many receptors we got? 1, 2, sarca 1, 3, and here in the T-tubule 2. One is exchanger, another is ATPase. So total how many? Right. Out of this, which one are present in cistern Wall of the cistern, RYR and sarca, SERCA. And two are present in the wall of the T-tubular membrane, means sarcolemma. What are these? What are these? This is one. DH. What is this? DH? Dihydroxypyridine receptors. So, other than there is two other. One is exchanger, another is ATP, plasma membrane calcium ATPase. Clear? Okay. So this part is over. Now we have seen calcium ion has entered. Now what this calcium ion will do? I am coming to diagrammatically. What is this myosin? Myosin head. Myosin head, it always wants to bind with its binding site present in actin globules. So these are the active sites present in the actin globules. But the problem is, these sites are not open. They are occupied by the thread. What is this thread? Tropomyosin. So these active sites are covered by tropomyosin. And who helps in this process? Binding of the active site of actin globules, binding of by tropomyosin. Who helps? Troponin. Troponin is having one side trop T. So trop T is helping binding of tropomyosin with the active site. So if I want to open it, then I have to do one thing. I have to displace this thread from the active sites. How it can be displaced? This troponin is having trop C for binding calcium ion. So now this calcium ion will bind with the trop C. Binding of trop C will cause uncovering of active site, uncovering of myosin binding site of actin molecules. So as soon as calcium binds to trop C, what will happen? There will be opening of myosin binding site. Now myosin is ready. Myosin is ready. Myosin head is ready. It is always interested to bind with this its site, but it is not capable of binding. Why? Because the site is covered. This active myosin head, this myosin head in our video we will find one battery is there. Battery, symbol of battery, means this head is charged, having some energy within it. Plus it is having ATP and PI. We'll see the movie. We will see the this animation. So myosin head, before its binding to the active site, it is having what? ADP and PI in the light meromycin. And here is the ATP binding site. So ATP is not there. So in that place, what is there? ADP and PI. Plus the head is having some energy. And this energy will be needed for swivel action, for power stroke, making power stroke. So it is ready to make power stroke. But before that, it has to bind to the myosin binding site in actin, G-actin. So what is there? Now, before it becomes bound to this, it is having ADP and PI plus energy. It is ready with it. So when calcium ion is released, or calcium ion became bound to trop C, then what will happen? This thread will be removed from there. Now this myosin binding site is open, so myosin will come and bind. Immediately what will happen? ADP will be replaced and phosphate will be released. Initially what was there? ADP and PI. When they are released from here, ADP and PI is released, then these are having energy. With this energy, it will make a 45-degree angle sweep. 45-degree angle sweep. So it was bound here. So if I sweep it, this will move. Similar, myosin is there in this side. They will also attach to the other side. So both actin will be coming closer. Same thing will be happening to the opposite side. M band. This side myosin is there. This side also myosin is there. So this side is pulling the actin. This side also will be pulling the actin. As a result, they will be coming closer. And in between the actin filament, there is a gap. This gap will be reduced. This gap will be reduced. As long as calcium remains bound, what will happen? It will make a swivel. A new binding site will come. So again it will go back to its original position and bind another site and make a power stroke. Before every power stroke, because for making one power stroke, it has utilized the energy. For next power stroke, it must have energy. How it will get this energy? ADP PI replaced. Now the myosin head is having a site for ATP binding site. Now ATP is stored in the sarcoplasm, they will attach. And this ATP will be now broken. What will be the result? If ATP is broken, what we get from ATP? ADP, PI plus energy. Now this energy will be stored again in the head, myosin head, and ATP and PI will remain bound. So it was the initial stage. So now it has affinity for myosin binding site. So now it will bind with the myosin binding site. Again, this phosphate and PI will be removed. There will be power stroke. Again, ATP will bind. So this will release from the site. After making power stroke, ATP will be broken, hydrolyzed. So PI and ADP will remain bound to it, and the head will be having now energy. So again with this it will bind, and this way it will be making this power stroke as long as calcium is bound there. When calcium ion gradually release of calcium ion within the cell comes to a specific value, then what happens? those receptors who are responsible for uptake of calcium ion, they will become activated. They will become activated. As a result of these, from sarcoplasm, calcium ion will be again entering to their original position. So, the calcium ion that was bound to the trop C will not be there. It will be coming out from there and will be entering within the sarcotubular system and into the cistern. So, no calcium ion, no more binding. Again, the active site will remain covered. So, myosin head cannot combine it. So, there will be relaxation. When ATP binds to myosin, it is hydrolyzed. With water, ATP is hydrolyzed. So now myosin will having ADP plus Pi plus energy. So now it has got affinity for actin. Now it will bind with the actin and form a actin-myosin cross-bridge. Okay? Now Pi and it will be revoked and again actin-myosin will be there. After some time, again ATP will bind. Myosin will bind, myosin will be getting. So this cycle will be going on. You might have heard the name of a condition called rigor mortis. Can anybody explain what is rigor mortis? Body becomes stiff after death, isn't it? Immediately after death, it will be okay. The body is flexible. But after 4 to 6 hours, the body gets stiffening. When you will find some dead body, if you find the body is stiffened, that means death has been occurred at least 6 hours before earlier. And this stiffening will continue for 12 hours. This stiffening will be present up to 12 hours. And after 40 hours, the stiffening will be completed. Total stiffening will be there up to 12 hours. And after 40 days, it will maintain, the stiffening will be maintained for how many hours? 40 hours. And after 40 hours, again we will find there is no rigidity. It is now flexible. What happened? What happened? The ATP, when myosin head became bound to actin. Okay? For its removal, what is needed? ATP is needed. It is now bound to the active site, bound to the myosin binding site. For its release, had to be released, no? It needs what? ATP. After death, no ATP. So this will remain fixed. Muscle will remain in contractile state. No release. So, and it's before that, immediately after that, there is brain death, there is or cardiac, no cardiac activity. But the cells are still working, that may have still some ATPs. So they will be utilizing those ATPs. So ATP remain bound there. As a result, muscle will be released, but after that some time there will be no ATP, so it will no fresh ATP will be coming to bind with it, so it will be remain, the myosin head will remain attached to the actin. So there is muscle is in the contractile state, so there will be stiffening. And what happens after 40 hours? After 40 hours, the cell undergoes putrefaction. उसका पचन हो जाता है. It gets, there will be apoptosis. The cell will die. There, everything will be, putrefaction. There will be putrefaction. So because of that, everything will be disintegrated. The muscle fibers, everything will be disintegrated. So no more contraction. It will be under relaxation. So this is very important when you will be doing post-mortem. You have to mention when death, at least death has occurred this much hours back. So this part is important. RY receptor, DHP receptor, when action potential came here, it is displaced. At the same time, this one is also displaced. So calcium ion enters, calcium ion enters. I think now you will be understanding this video properly.
In this video, we will see the physiology of muscle contraction. We will see excitation-contraction body, and then myosin cross-bridge cycle that ultimately produces.
What is this receptor? DS and this RY ryanodine receptors. So calcium ion.
Then watch it first if you haven't already. On that note, let's get started with a quick revision of the anatomy.
Sarcoplasmic
Contains myofibrils, sarcoplasmics,
T-tubules,
sarcoplasmic reticulum.
and these are the sarcoplasmic reticulum.
Myofibrils are cylindrical and arranged.
Jet lines, myosin, and these are the actins.
Myofibril is a chain of regularly repeating units called sarcomere. Sarcomeres contain thin filaments and thick filaments. Thin filaments consist of actin, tropomyosin, and troponin. Filament actin makes the backbone of the thin filament. Tropomyosin and troponin are regulatory actin-binding proteins. Under the resting condition, the tropomyosin covers active sites on actin molecules.
Blocked.
So that was thin filament. Thick filaments interdigitate with thin filaments. They consist of many myosin 2 molecules. Myosin has a tail where all the myosin molecules are connected and a cross-bridge region that extends out. Cross-bridge has a globular head and arm. So these are myofibrils. Now surrounding myofibrils is T-tubule sarcoplasmic reticulum system. T-tubules are imaginations of plasma membrane. And sarcoplasmic reticulum is a special type of endoplasmic reticulum. This is the longitudinal cross section. These are T-tubules. The sarcoplasmic reticulum is made up of longitudinal tubules that make most of the area and terminal system that are close to the T-tubules. This assembly of T-tubule in the middle and two neighboring terminal system is called triangle. This was all inside the cell. On the cell membrane, we have neuromuscular junction that increases the actin of terminal and post-synaptic membrane, which is basically muscle plasma membrane. This was called the anatomy. Now let's see how muscle contracts. First of all, the stimulus for contraction comes along a nerve and reaches the neuromuscular junction. After all the units of neuromuscular transmission, which are already covered in a separate video, and action potential is generated as opposed to acting membrane. The action potential spreads along the membrane. Along ABUs, it penetrates deeper into the muscle cell. Now let's go to the transmission where the next magic happens. As we have seen, the action potential passes through the ABUs. Now here we have another type of calcium channels on the membrane.
DSP. This is DSP here.
Causes have to open. This causes an influx of calcium
Open.
into the cell. See, the T-tubules are imaginations from cell membrane. So the fluid inside is in fact extracellular fluid, and the calcium entering through these channels is extracellular calcium. Now these calcium channels make physical contact with calcium.
What is this channel? What is the name?
So the opening
RY?
calcium channels
Yes, sir.
mechanically opens calcium in these channels also. This causes diffusion of calcium from the sarcoplasmic reticulum into the sarcoplasm. Thus, this calcium is coming from cells whose storage in sarcoplasmic reticulum. In a nutshell, action potential causes calcium entry from extracellular fluid as well as sarcoplasmic reticulum and increases cytosolic calcium concentration. Now, the calcium spreads into the sarcomere.
Now,
The tropomyosin molecule is covered in the binding site contacting molecules. So myosin is not able to bind to actin. Now the calcium has just come binds with its friend tropomine. This binding causes
drop C plus calcium ion.
tropomyosin into the groove in between two strands of actin molecules.
Now this thread shifted from here to there.
Now uncovered.
Now it is open.
Myosin to bind here
Bind.
and continues the cross-bridge cycle. This brings us to the myosin cross-bridge cycle. Basically, it is this type of repetitive movement of myosin head that pulls the thin filaments. Let's see what happens exactly in detail. We will start with the state of myosin head just before binding to the actin. It is in line with the arm, and at this point, it is bound to ADP and a phosphate molecule.
ADP, PI plus energy.
All is wants to bind the actin, but
This is energy.
binding was prevented by tropomyosin, which was covered in the binding site. But now, as the site is exposed, the head can bind with the actin.
Now ATP will bind.
Where the head
After
and
swivel,
head
now ATP will bind.
over the thick filament towards the center of the sarcomere. Phosphate is released during the power stroke, and ADP is released right after it.
Power is also exhausted.
Molecule binds with the head. This binding reduces the affinity of myosin for actin, so it detaches from the actin.
So ATP is again charging it for this section.
The released energy is used to bend the head back into the initial state. Likewise, another actin molecule and the entire cycle is repeated. This continues to occur till the site connecting is uncovered. The other head of the same myosin 2 molecule, as well as all the other myosin molecules do the same thing. In this way, thin filaments are pulled over thin filaments. The same thing happens at the opposite end also. Thus, the pulling shortens the sarcomere. A similar shortening of sarcomeres reduces the length of muscle fiber and thereby muscle, and this produces muscle contraction. Now, few points regarding the length of different bands. See, the A band is the region of thick filaments, and I band is region of thin filaments without overlap.
M will be reacting.
We have sodium calcium exchanger and plasma membrane calcium MBBS that pumps calcium out of myosin into the extracellular fluid. And on the membrane of cytoplasmic reticulum, we have sarcoplasmic and endoplasmic reticulum calcium ATPs that pumps calcium back into the sarcoplasmic reticulum. Both of these exits reduce cytosolic calcium concentration.
Now calcium will be removed.
This moves the tropomyosin back to the resting position.
ATP binding site covered.
That's on the actin molecules.
No more binding.
So now the myosin head cannot bind with the actin and the process stops. ATPs are available. The head takes this position. It is a common position in which the head is ready to bind with the actin and pull it as soon as the binding site is exposed to the next strand. It is like walking a gun to make it ready to fire. So this was all about the physiology of muscle contraction.
Okay, now we'll discuss. That is over. I think that you don't have any doubt regarding mechanism of muscle contraction. Okay, structure and function. So you have to explain the structure of myosin, what is there in myosin, what is there in actin, so you have to explain. EC coupling or simply mechanism of muscle contraction. So you prepare for any type of question, long type from this. Okay? So now we'll discuss, these are short questions. These are what? Two type of muscle contraction is there. One is isotonic, another is I'm going to contract my biceps. Okay, it is contracted. So this contraction, what happened? It moved this arm, this here. So there is displacement. Because of contraction, they are pulling this weight to a new position. So there is a work. Displacement, force generated, so force into displacement, so work is done, being done. This sort of contraction is called isotonic contraction. So when I contracted up to this muscle is contracted, but within this muscle tension is same. More contraction, more movement, still the tension is same. Isotonic. So during contraction, no increase in tonicity, no increase in muscle tension. Only there is shortening. Understood this point? This type of contraction, I am lifting this to this new height. So this is what? Isotonic contraction. Now I am doing another type of action. Muscle contraction. I am pushing this wall. Pushing this wall and trying to send it back. I have exact muscle contraction was there, but there was no shortening. The only thing, tension has increased and no work has been done. No work has been done. So these are the two main type of muscle contraction. In gym, you can do isotonic as well. You will be running on a treadmill, you will be cycling. These are all isotonic. But when you will be lifting or holding some weight, then these are isometric. Short question. This much is enough. So you write it, copy it in your book, in your notebook. And isometric. I explained what is isometric. See the difference. These are the difference. Go through it. Length shortens in isotonic, no shortening. Tension remains same, but here tension increases. Movement occurs, no movement. Load is this. Load is displaced. Load is not displaced. I could not move the wall. External work done, but here no work has been done. More efficient, less efficient. Your car, if you are trying to move it, But acceleration, you are making a lot of acceleration, but car is not moving. So no work is done. Less efficient. Using less energy, if you perform if you are capable of doing more work, then it is called more efficient. Okay, so the efficiency is less. Example, lifting a load, pushing a wall. So now it is clear to you what are the different types of work. Positive work. This is a stair. I am lifting my body weight here. So there is displacement. I can measure the work. So this is a positive work. But I am coming down, favoring the gravity. So this is a negative work. While you will be sitting, some muscles, extension muscles, they are not contracting, they are further relaxing. So no contraction, only stretching is there. Muscle is elongated. Then only you can sit. This extension muscle, using this extension muscle, you could sit, you could able to sit. So there is no contraction. So this sort of work is called negative work. Example, and this will take another class. Now class is over and already 20 minutes. So this is also important question. I am doing it. So lot of ATP is needed. Where from this ATP will become? So that I will be explaining. Moreover, after that, what is retained in this unit? Hello? Some properties of the muscle, like summation. What is summation? So you have to explain it. You can expect this question as short question. Another point is graded work. Graded work. Gradation of work. Gradation of exercise. Gradation of muscular activity. In most of the books, these are not available. These are not available. But in your new curriculum, it has been added. But we know how these are graded. I'll give a note. So you just copy it. Since it is newly added in 24 syllabi, so question may come. Like newly added item is brain gut axis. So you can also expect it. That's why I've given this question. Now, how these are graded? So this is the sedentary lifestyle, means mild physical activity, moderate physical activity. In the morning, you go for a walk. In the afternoon, you dance or do some games, perform some games. So these are moderate. So light, moderate. And these are gradation. And above this, one rickshaw puller. Every time he has to work. It's heavy. And another is strenuous. Pulling body weight. Or running 100 meters or marathon running. Or working in a field. Or breaking stones. So these are strenuous exercise. This type of exercise cannot be continued for more than 2 hours. Then he or she has to take rest. Then only again he can repeat the process. So these are strenuous. So how many gradation is there? Mild, moderate, heavy, and strenuous. What are the criteria of classifying these? In the next class, I will explain. One is criteria is given by who? Who means World Health Organization. Another classification is made by the exercise physiologist. Okay, so both of these classification we will undertake.